Well, dear students, hope so you're doing well. In my today's videos, I'm gonna open the Pandora box of Euchromatin and a Heterochromatin. So let me tell you one thing that chromatin is a uncondensed form of a DNA. You can say that the chromatin is basically when you have a thread which is not wrapped over in a proper way of wrapping. Improper way of wrapping is basically chromatin. If the DNA is being not wrapped properly, it is just there in a kind of a there in its condition of shape, then you can say it's a chromatin. Otherwise it is a chromosome. Soma mean a body. When it just took a proper shape, then you can call it as a body. It's a chromosoma. But the story comes here that what is euchromatin and heterochromatin? Scientists have figured out that if we have a kind of a chromosome, when it is there in a form of chromatin, you can say at centromere, this is called the centromere, this position, the chromosome never goes for DNA to RNA function. That is called the transcription. If DNA is not performing its central dogma or RNA function, DNA is not making RNA, its machinery is not being used, then this thing, the part of the DNA, the part of the chromosome, the part of the chromatin is a euchromatin. But when you see that certain most parts of the chromosome are being used for the production of RNA, that is called the RNA synthesis, and later on these RNA goes out from the nucleus, and after going out from the nucleus they're gonna go towards the ribosomes, and that they're gonna make some protein synthesis. They go for some kind of phenotypic expression or phenogenesis, you can say, is a euchromatin. But when the chromosome is not in a condition to give its RNA, or somehow some circumstances or more condensation never allowed it to get into RNA function. The DNA stain is quite dark, and there's a lighter stain for a DNA. You know, if you have a DNA, there is a darker stain and a lighter stain. The darker stain is because of the part is not being used, and the lighter stain parts are being used. The parts which are used, they are euchromatin, and the parts which are not used, they are basically the heterochromatin. And you can call these heterochromatin in a two respected manners, that you can call them as a constitutive heterochromatin or facultative heterochromatin. So what is constitutive and facultative chromatins? They are basically when you say that there are certain region on a chromosome which is there in the form of centromere, the part of the centromere which is not usable, genetically inert part. This is constitutive heterochromatin. And when the sex cells are there, the sex chromosomes, yeah, sex chromatin, in that part, the DNA, the chromosome part which is not functional, that is the facultative heterochromatin. And in that, you can say sex chromatin is going to be late replicating one. Sometimes it in a future it replicates itself. And I hope you're getting my point. Euchromatin, which is being converted into RNA. The DNA machinery has done its job and RNA is being produced. Heterochromatin, the DNA is not functional, and it is not going to make allowance for the RNA to produce or synthesis of RNA. So in easy words, this is the basic points which I told you. I hope you're getting one. Thank you for watching.
Thank you for watching.
GS Academy.